episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome everybody again to another episode of our show. We're bringing you another fascinating guest today who is helping to create a better tomorrow for all of us. We are continuing our virtual trip around the world. We are headed over to Japan today, and we have the honor of meeting with Dr. Tohru Minamino, who is professor and chairman of the Department of Cardiovascular Biology and Medicine at Juntendo University Graduate School of Medicine, and he also serves as director of the Cardiovascular Medicine Center there at Juntendo University Hospital. Dr. Minamino received his medical degree from Chiba University Graduate School of Medicine, his PhD from the Faculty of Medicine at the University of Tokyo. And he is a medical cardiologist and research scientist who is very interested in focusing on the molecular mechanisms of aging. He started major research in this area, focusing on cardiovascular aging when he was at Harvard Medical School back in the late 90s. And his research interests have currently been growing in the biology of aging, including both metabolic pathways of longevity and senolysis. He has published more than 100 papers, including those in Nature Medicine, Cell and Cell Metabolism, and the Lancet, and has won several awards over the years, including the Satoh Memorial Award in Japanese Circulation Society and the Erwin von Bälz Award, first prize. A lot of very interesting things he's doing nowadays that have been in the press. So we welcome Dr. Tohru Minamino to our show today.
Thank you for your kind introduction. My name is Tohru Minamino from Juntendo University. And I don't know exactly what this, the project is. I mean, but hopefully I could introduce myself as a, cardiologist as well as physician scientist. And actually, I initially wanted to become a family doctor, not basic researcher or cardiologist, because my grandpa and my, many relatives, are sort of family doctors. They, so are, the patients who are living in the regional regions, and who care, they care the patient with a common cold or and what such a very common disease. But I was attracted the cardiovascular medicine because of its clinical dynamism. And, usually we took, 5 years residency to become a cardiologist. And during the time, I experienced several patients with familial cardiomyopathy. So at that time, it was unknown, the genes that account for such familial disease. So I was very interested in the such identification of such responsible genes. So I then become interested in the basic research at that time. And initially, I graduated from Chiba University, but in the Chiba University, I could not perform such basic research, especially in cardiovascular field. So I actually visited the University of Tokyo, especially Professor Yoshio Yazaki's group. who are, focusing on the basic research in the cardiovascular field. Actually, his laboratory is a sort of mecca for molecular biology.
Sure.
In the cardiovascular field. Actually, you may know the Issei Komuro and other famous cardiovascular biologists, especially as a basic researcher. And actually, initially, I planned to join the heart failure group because I wanted to study about the familial cardiomyopathy. But eventually, I was assigned to a vascular biology group, which was governed by Yuki Kurihara, who are now professor of University of Tokyo. And I was focusing on the endothelin and its converting enzyme in vessels. And I studied, such vascular, biology. And I was very impressed by several, things. One of which is, the space for, research. I mean, study research is very small, and I considered that Professor Yazaki is very famous, professor. So I thought that the space must be huge, but very unfortunately it's very small. But it's but it was very impressed. But initially, I was asked by Professor Dr. Kurihara that Dr. Tohru, where would you like to have your drawers? So what? So I believe, I believe that I would be given one space, table, but just they provide me very small drawers. So I was very shocked, but it's not a problem. And many maybe about 8 or 10 people researchers are working very hard in a very small space of the Tokyo University, and from early morning to midnight. And I was also working hard actually, and I completed my first research on endothelin converting enzyme in atherosclerosis for the initial 2 years or 3 years, and it was my actually thesis, and I could get a PhD degree in the University of Tokyo. So then afterwards, I thought, or I tried to find out my own field. And at that time, the internet service is not available well,
Sure.
which is about around.
It's very slow at that time.
Yeah, right. So, and I, frequently went to the library, and I tried to find out, any interesting topic. And finally, I found out the one review paper which described about the methodology by which telomerase activity could be measured. So I don't know. I was not familiar with telomerase at that time, but I was very interested in the telomere and telomerase. And actually, at that time, telomerase activity could be only detected as an activity.
Sure.
But nobody knows about the molecules of the telomerase. But yeah, I thought that it was very interesting to me, and so I decided to study telomeres and the telomerase in my field, especially in the blood vessels. And actually, I hypothesized that once cellular aging per se can promote vascular aging.
Mm-hmm.
That was my initial hypothesis, which is known as a cellular senescence hypothesis, I think. Then I then thought that I should go elsewhere, especially I should go abroad or to the United States or to the Europe to study about this new topic. However, at that time, telomere and the telomerase biology was active in cancer field.
Yep.
So, but I would like to, study about this topic in the cardiovascular field. Mm-hmm. So I was very concerned about my, identity as a, researcher. If I go to the laboratory focusing on the cancer biology, because I was a cardiologist. So I decided to visit the vascular biology laboratory, which was governed by Professor Colin Barnes at Harvard Medical School. And she was focusing on the hypoxia and angiogenesis, which is also, very interesting topic to me. So I decided to join that laboratory. But when I took an interview with her, I asked her whether, I could also study about telomere and telomerase as a, second topic. Mm-hmm. And fortunately, she agreed with my suggestions. So then, I started to study both, hypoxia topic and vascular aging topic. And
if I could interrupt for 1 second at this point, because I think this is a very interesting inflection part of the story, because it is here, as I was reading a little bit about this history, you begin to publish at this point when you're at Harvard on things like hypoxia, and vascular smooth muscle cells and telomerase activation. Right. You published on endothelial cell senescence. This was all back in 2000, 2001, and now we're 20 some odd years later. What was sort of the response at that point? Because this was a couple of decades ago, and you're working really on the forefront of topics that are very hot nowadays in 2022. What was going on in the lab at that time? Like, what did they say after you started publishing this stuff?
Yeah, actually, so if when I submitted the paper, for example, on telomere and telomerase, so many, reviewers had a comment on that, which is like that. Why, do you, need to study about telomeres and telomerase in vascular biology? So, which was very shocked to me, right? Because I believe that, telomeres and telomerase or cellular aging pathway play an important role in vascular aging, but many people did not recognize as a important part of biology, vascular biology. So that's why I could not get any publications during the about 3 years at the Harvard Medical School. So I actually worked very hard, and I got some results, but nobody accepted my research suggestions. So even in the case of the American Heart Association meeting, they do not accept it. My findings as a topic. So I almost had given up my field. So I actually came back to Japan.
And so this is another interesting as I said, once again, reading about the journey. You come back to Japan, you're at Chiba University for a bit, and you're continuing some of this work, but then you a few years after that you were asked to preside over the Department of Cardiovascular Biology and Medicine at Niigata University. And this is where you start publishing and really making the full connections here, because you start publishing at this point on not just senescent cells accumulation in the heart, but also in fat. And now you're making some very interesting cardiovascular, diabetes, adipose tissue connections. You start publishing on adipose tissue inflammation. You make a really, you publish a paper on semaphorin induced inflammation, which we talked about semaphorins on the show actually before, but mainly from the neurological side of semaphorins. And this was another fascinating paper of yours in dietary obesity. Talk about this era, because I think this is, and we'll get into senolytics, senolysis in a little bit, but talk about this point, because I think this is a very important point in your journey where you start making the, what I'll call the full circle connections between what you were doing.
Yeah, because, when I was working at the Harvard Medical School, nobody, appreciate my research field. So I, considered that I need to publish paper which clearly indicate that cellular aging pathway play a very, very important role in cardiovascular aging. So for this purpose, I actually focus on the P53, which is well-known the tumor suppressor gene, but also known as a promoter of cellular senescence. So if you inhibit P53, you may be able to inhibit cellular senescence, right? So that if I can prevent, for example, diabetes by deleting P53, I can clearly indicate that causal relationship between cellular aging and various age-associated disease. So I therefore then started to focus on the in vivo studies, especially in which I manipulate P53 status in various tissues. And show the causal relationship between P53 or cellular aging and age-associated disease by tissue-specific P53 deficient mice. For example, if you inhibit P53 in the vasculature, you can prevent high-fat diet-induced vascular dysfunction, which is associated with accumulation of cellular senescence. You can also inhibit or prevent the development of diabetes by deleting P53 in adipose tissue. Because the senescent cells, senescent adipose cells accumulate by high-fat diet intake or with obesity. So I really, initially focused on the, proof of concept for cellular senescence hypothesis in our field. So I successfully showed that important critical role of P53 dependent of cellular senescence in vasculature or for in the development of atherosclerosis. I also could demonstrate that P53 also critically involved in the heart failure development, or adipose tissue. P53 is also contributing to the development of diabetes. So, that was, done at the Chiba University as well as Niigata University. And I also noted that, one tissue aging could cause another tissue aging. Like, for example, if you introduce the cellular aging in adipose tissue, you may also observe the heart failure development. So I believe that, organ aging is closely linked to each other. So I think one organ aging, if you could improve the one organ aging, you may be able to improve the whole body aging. So that's what we are working on this field at Chiba University as well as the Niigata University. But so then finally, we have to move to the therapy, right?
Yep.
But in the experimental model, you can inhibit P53 to prevent cellular senescence and improve the diabetes or heart failure or atherosclerosis. But in the clinical setting, you cannot inhibit P53, obviously, right?
Sure.
So, and in 2011 to 16, our community, aging community, found out that elimination of senescent cells could be novel strategy to inhibit aging. So then we, our group also moved from the, pathology to treatment. And, initial study showed that the genetic deletion of the genetic elimination of senescent cell can improve the organismal aging in various conditions. And afterwards, Dr. Cochran group showed that, several compounds, which is known as senolytics, could remove specifically senescent cells and prevent or improve the pathological aging phenotype. But I was concerned about the, off-target effect because, most of them are, a sort of, a kind of the anti-cancer drugs, so that it may also affect normal cells, right? And actually, the one of the most famous senolytics, ABT, is anti-cancer drug, which affect the bone marrow cells. So we should. I think that I should take another approach to develop senolytic treatment. So in 2014, around 2014, we tried to identify the surface molecules, which should be expressed by specifically by senescent cells, especially senescent endothelial cells. And we searched various omics data, including ours, and we found out several molecules which are specifically expressed by senescent endothelial cells, and which is located on the cell surface of endothelial cells. And I thought that we may be able to target these molecules to eliminate senescent cells specifically. So then we first of all try to characterize such specific molecules. In this case, we first of all working on the GPNMB, which is which is not famous molecule actually, but which is very specifically expressed by senescent endothelial cells.
Yeah, if you don't mind me interrupting you here, because I think this is another really fascinating part of the strategy, and I'd just like to go a couple minutes into this, if you would, because just for the audience, GPNMB, glycoprotein, non-metastatic melanoma protein B, involved, on the surface of cancer cells, but at the same time, a very unique biology in terms of angiogenesis, matrix metalloproteinase dynamics. Talk about that a little bit, because I think that's really crucial in understanding sort of the targeted nature of what you're doing here and how you thought through, okay, here's something that's more, when you think cancer invasiveness related, but when we think of how cancer invades and vascularization, talk about that connection, because I think that is so important to connecting back to your cardiology work.
Yeah, actually, recent our publication in the Nature Aging do not describe about any characterization of GPNMB, but we know that GPNMB is very, very unique molecules for cellular senescence, and we believe that GPNMB is specifically expressed by very, very, very senescent cells. So because our study indicate that GPNMB play an important role in lysosomal integrity, which is unpublished. And recent paper describing about GLS, glutaminase, activity in cellular senescence which is published from Tokyo University, Dr. Nakani's lab. They also noticed that lysosomal integrity is very important role in senescent cells. And actually GPNMB, we found that the GPNMB is a positive regulator for lysosomal integrity in senescent cells. And actually they act as a survival factor for senescent cells, which means it's like a cancer antigen, which accelerate cancer cell survival. So according to our recent data, we speculate that GPNMB is upregulated by lysosomal stress, which is common phenomena in senescent cells, because senescent cells cannot divide, but they produce a lot of junk,
right?
Which should be somehow managed by lysosomal or other organelles. So and upregulated GPNMB contribute to survival of senescent cells. So I think that senescent cells expressing high level of GPNMB is very, very malignant one, which secrete a lot of SAS factor, pro-inflammatory cytokines. So I think the GP targeting such molecule is reasonable to improve the pathological aging. So this molecule is not simple, senoantigen, but they, is a sort of marker for malignant senescent cells. So that if you eliminate GPNMB positive cells, you can get enhanced, results. Improvement. Yeah, that's what we are now speculating as for GPNMB. So, yeah.
And continuing along that, you published recently this paper. You're famous around the world in the press a couple of weeks ago. Senolytic vaccination improves normal and pathologic age-related phenotypes, increases lifespan in progeroid mice. Just talk a little bit about that paper, if you would, some of the sort of feedback that you've gotten, and then I'd like to. As a subset of that, obviously, this is an amazing set of experiments you did here. A little bit, what's next in the sense that you mentioned senoantigens. We're in the very early days of really knowing you've highlighted this one in terms of GPNMB. Is the plan to look for more senoantigens? Is it to further modify this specific vaccine? Talk a little bit about what is going to be happening next after this, but please take the floor back.
Okay, great. Okay, so first of all, in our recent publication in Nature Aging, we identified GP as a senoantigen, especially in the senescent and endothelial cells. And this molecule are markedly upregulated in the senescent endothelial cells as well as some immune cells, actually. And we also found out that this molecule is markedly upregulated in human samples, aged samples. And we initially, to get the POC, we first of all established the genetic model that can eliminate the GP positive senescent cells, by injecting for example, diphtheria toxin. And we actually observed that elimination of GP positive cells can improve diabetic condition, atherosclerosis. So we then moved to the next step in which we are trying to develop the clinical application.
Mm-hmm.
And we decided to utilize the peptide vaccination strategy because it's very easy and to develop. And we designed several, peptide vaccination.
Mm-hmm.
And fortunately, we could one, candidate. And if you treat the mice with a peptide vaccination, and you can get the increased antibody titer against the GP after 2 weeks after vaccination until, let's say, to 20 weeks, maybe. So which is nice. And we first of all examine the effect of this vaccination in the diabetic mice, and we found that its improvement. And we also treated atherosclerosis prone mice, ApoE knockout mice, with this vaccination, and we could attenuate atherosclerosis. And we also found that if you treat the aged mice with this vaccination, you can preserve frailty. I mean, you can improve frailty. I mean, you can preserve the exercise capacity until, old mice. And finally, we could observe that this vaccination could extend the lifespan of premature aging mice. So what is next? Actually, we so far we found out the several xenoantigens, which were specific for senescent endothelial cells. I could, actually go for it, but I also, I'm interested in the clinical application, obviously. So, there are several problems regarding the clinical development of the vaccination. Because it's, in our case, peptide vaccine, right? So it's a little bit tricky to apply for the human disease. So I decided to develop the antibody strategy. So actually, we are now working on the development of the human antibody against GP. And we have already cloned, gotten the several, couple of clones that could specifically act with GP. So I may be able to develop, first of all, imaging technique to detect GP positive senescent cells by using such antibody, like a as a PET probe. So next, I wish to develop the treatment strategy by using those antibodies. And we are now collaborating with several groups who are working on the radioisotope, for example, or several, drugs that can kill the cells, like a cancer drug. So that we can eliminate specifically GP positive malignant senescent cells by using such antibody conjugated isotope, or antibody conjugated drugs, or I may also antibody pathway. If antibody pathway have ADCC activity, I can use antibody pathway as a drug to eliminate senescent cells. So that's what we are now working on in this field. But we also try to examine the elimination of senescent cell by targeting another xenoantigens as well.
Excellent, excellent. And I just want to mention, you also you've quite recently in a new book entitled Cellular Senescence and Disease, you published the chapter on vascular diseases, atherosclerosis, and atherosclerotic cardiovascular diseases, is chapter 9. And once again, you're sort of looking forward at this, and you mentioned in sort of in the abstract here about as you mentioned, the senescence associated circulatory phenotype, or the SASP, or a lot of the junk that these cells give off. I mean, I obviously have a lot to do, and there's a lot going on your plate right now, but any thoughts of sort of using some of these antibody approaches to also soak up SASP and SASP junk alongside senolysis, or is that another group working on that?
Yeah, actually targeting SASP is also another strategy to eliminate, to improve pathological aging phenotype. But I am afraid that in that case, we need to treat the patient, continuously treat the patient with antibody, right? But in the case of the antibody conjugated some drugs to eliminate senescent cells, you can just treat once, maybe.
Got it. Okay. Yeah,
right?
no, makes sense.
It's a big advantage, right? As for the treatment. So another issue I would be concerned about SASP targeting treatment is that if you suppress SASP, you may also accelerate accumulation of senescent cells, right?
Interesting. Because
SASP is originally secreted by senescent cells to promote elimination of themselves, right?
Got it.
So
Got it.
If you inhibit SASP, you may inhibit elimination of senescent cells. So I am afraid that part. So
that makes sense.
Right.
Makes sense.
But I also am interested in the identification of the mechanism by which immune system somehow is impaired as for elimination of senescent cell.
So sort of immunosenescence.
Right. So it's like PD-1. I mean, actually, some group have shown that the PD-1 or PDL-1 is play an important role in energy for senescent cells. So
interesting.
I think there is another mechanism by which the senescent cell can be survived or can be can escape from the immune system to be survived. Yeah, that's what another. I mean, that would be another interesting topic. To investigate.
Doctor, what is the current in 2022? What is the current aging biotechnology research space like in Japan right now? Is it a very active space? I mean, in the United States, it's taken off in the last couple of years. What are things like in Japan right now?
Yeah, actually, obviously we are delayed. I mean, as for the development of the aging strategy treatment, but recently the government tried to support our field to develop novel strategy to improve the aging phenotype, because our society obviously becoming our age. About 30% of population is now over 65 years old now in Japan. So it's one of the top leader of the aged society. So I believe that various budget has been developed in Japan for the aging research now, and many research group now try to commit to this field. So I think the aging field in Japan become more active within a couple of years, and I hopefully to establish venture company, obviously, to facilitate the such activity. I mean the promote activity. So yeah, I now becoming optimistic. So as for aging research in Japan.
Excellent. Excellent. And one last question along those lines. There was a conference, I think it was back in March, that you presented at, and somebody else that I guess gets a lot of press over here in the U.S. Is obviously 2012 Nobel Prize winner Shinya Yamanaka, and on induced pluripotent stem cell fame, did a little work in the cardiovascular space as well. Just like to get your top line on cellular reprogramming as it pertains to cardiovascular health. Are you doing anything? I mean, I don't know if you're partnering up with Dr. Yamanaka at all, and if he's confidentially able to talk about it, but give me your top line on cellular reprogramming, specifically as it pertains to the cardiovascular system.
Yeah, that is another interesting topic actually, and I obviously I do not focus on that because too much.
Sure, I understand.
Yeah, but reprogramming can recover DNA damage actually, right? So it's very, very fantastic methodology to recover the aging phenotype. But my concern is about the tumorigenesis.
Sure.
Right. So I guess the elimination of senescent cell is more reasonable to apply for the human settings. But in the experimental setting, I believe that the Yamanaka factor is very interesting to be investigated. So as to apply the recovery of aged cells.
Yeah,
right. So, but another interesting topic is sirtuins that that may be able to partly recover the DNA damage, right? So if you combine sirtuin stuff and the senolytics, you may be able to, improve the, restore DNA damage, but also eliminate very, very, bad damaged cells. So that's would be feasible, maybe for human disease condition, I guess.
Yeah, it's I just brought that up because I think we forget that cardiovascular disease is still killing 1/3 of the world every year, and it's important to see thought leaders like yourselves and Dr. Yamanaka and so forth really working on these different strategies. But you definitely are at the right spot, and clearly it's been a very impressive history that you've walked us through. While I have you still, any other big news that you care to mention for 2022, and obviously nothing confidential, but anything that you want to mention that I haven't asked about, please, I'll give you the floor for the final message.
Okay, so I guess it's not huge message, but I hope that our findings could be applicable to human disease, and especially if you look at cancer cancer treatment development, and it's there are several choices, right, to treat the cancer patients, and depends on the cancer types, right? So I believe that senescent cells also are heterogeneous, maybe.
Sure.
So I guess that we can identify several, for example, senoantigens or target to treat senescent cells. So for example, if you treat the cardiovascular disease, you may be able to, a couple of senoantigens to treat, but also you may also be able to select another senoantigen to treat kidney disease, for example. And you may be able to identify accumulation of specific senescent cells that specific, specific senoantigens by PET for CT, for example, by using a specific antibody. In that case, you can choose, right? Specific treatment like cancer. So I believe that our next stage would be, we need to develop the individual, treatment for each conditions. And I believe that we can do that, I think, in the near future. So it is my hope.
Yeah, I definitely see it happening. And yeah, the strategy that you've outlined is elegant, targeted, and it is definitely moving the field, I think, in this really personalized, as you were saying, direction. So it's going to be an exciting next several years to continue to watch this work and your work in the space. And this is still a major unmet medical need all around the world. So it's exciting to see thought leaders like yourselves working on it and your team. For everybody that is going to be listening to this episode of the podcast across the different networks or watching on the YouTube channel, once again, we've been speaking with Dr. Tohru Minamino, professor and chairman of the Department of Cardiovascular Biology and Medicine at Juntendo University in Japan, also director of Cardiovascular Medicine, Juntendo University Hospital. Doctor, it's been a real pleasure listening to your story. I want to thank you for taking the time out of your schedule to, and there's the evening there, to talk to us. Thanks, obviously, for everything you're doing. And as we say on this show, thank you for helping to make a better tomorrow for all of us. Really very impressive story.
Thank you very much. Thank you for your, your invitation to this program, and thank you for your listening, everyone.